Hello everyone, I am Nikita Tajne and in this today's topic we are discuss about the cellular receptors. So first we define about what are the receptor. It is defined as the macromolecules or the binding site located on the surface or inside the effector cell that serve to recognize the signal molecules or the drugs and initiate the response to it, but itself has no other function. Then cellular receptor are of four types: G-protein couple receptor, enzyme link receptor, nuclear receptor, and ligand gated ion channel receptor. So first we discuss about the G-protein couple receptor. These are the large family of the cell membrane receptor which are linked to the factor through one or more GTP activated proteins, that is G proteins, for response evacuations. All such receptors have a common pattern of structural organization. The molecule has a 7 alpha helical membrane spanning hydrophobic amino acid segment which run into the 3 extracellular and 3 intracellular loops. The agonist binding site is located somewhere between the helicals and the extracellular faces, while other recognition site formed by the cytosolic segments but bind to the G protein. The G protein flat in the membrane with their exposed domain lying in the cytosol, and the heteromeric in composition, that is alpha, beta, and gamma subunit. In inactive state, GTP bound to the alpha subunit at the exposed domain, and activation through the receptor leads to the displacement of GDP by the GTP. The inactivated alpha subunit carrying GTP dissociated from the other two subunit and either activates or inhibits the effector. Then we see about the second receptor, that is ion channel receptor. These cell surface receptor are called ligand gated ion channels, enclose the selective channels for the sodium, potassium, calcium, and the chloride ion within their molecules. Agonist binding opens the channel and cause a depolarization, hyperpolarization, changes in the cytosolic ionic compounds composition depending on the ion that flows through the nicotinic, cholinergic, GABA, glycine, excitatory amino acid glutamate, and the 5-HT3 receptor falls in this category. Then next we see the transferase JAK-STAT binding receptor, in which these receptors differ from the other receptors is not having any intrinsic catalytic domain. Agonist induced dimerization alter the intracellular domain conformation to increase the affinity for a cytosolic conformation to increase tyrosine kinase, JAK, that is Janus kinase. On binding, JAK gets activated and phosphorylate tyrosine residues of the receptor, which now bind to the another free moving protein, STAT, that is signal transducer and activator of the transcription. Then next we see the nuclear receptors. In contrast to above four classes of the receptor, these are intracellular soluble proteins. We respond to the lipid soluble chemical messenger that penetrate the cell. The receptor protein is inherently capable of binding to the specific gene but is attached to the proteins, and may some others prevent in adopting the configuration needed for binding to the DNA. When the hormone binds near the regulatory segment, carboxy the receptor, the restricting proteins are released, and the receptor dimerize, and the DNA binding regulatory segment located in the middle of molecule fold into the requested configuration. Then we see about the next receptor, that is. So thank you everyone.